Hello, future doctors. Welcome to memoneet. My name is Anupam Sinha, and I am working as biology subject matter expert at here memoneet. Today, I am going to discuss Botany Brahma's test series, that is PPT 11, and the topic is Principle of Inheritance and Variation. We will discuss section A, and we'll start with question number 101. Let's read what question says. The question states that Gregor Mendel conducted a hybridization experiment on garden peas for, and the options are 7 years, 8 years, 6 years, 5 years. It means this question is asking for how long or for how many years Gregor Mendel conducted his hybridization experiment. We all know that Gregor John Mendel is known as father of genetics, and he discovered the basic principle of inheritance, and for this he conducted a hybridization experiment, and he conducted it on garden peas for about 7 years, that is from 1856 to 1863. So let's read the option. The option first is 7 years, so this is the correct answer. He conducted that hybridization experiment for 7 years from 1856 to 1863. So the option number 1 will the correct answer for this question. Now let's move to the next question, that is question number 102. Question number 102 states that on the basis of observation on monohybrid crosses Mendel proposed, means by observing the monohybrid crosses, what laws Mendel proposed. So based on his observation on monohybrid crosses, Mendel proposed generally two rules, and he did it to consolidate his understanding of inheritance in monohybrid cross. And today these rules are called principle of inheritance or laws of inheritance. The first law is known as the law of dominance, and the second law or the law of segregation. Now let's read the options. The options are, first option is law of dominance. This is the correct. Second option is law of segregation. This option is also right. Third is law of independent assortment. This is not right. He given only two laws on the basis of monohybrid crosses. And fourth option is both 1 and 2. It means our answer for this question will be option 4. That is both 1 and 2. Now let's move to the next question, that is question number 103. Question number 103 states that the F1 resembled either of the two parents or was in between. If the F1 generation resemble both parents, it is the case of. So we know that when F1 resembles either of the two parents, then such situation is called dominance. And if the progeny or F1 result resembled in between, that it is known as incomplete dominance. And when the F1 resembled both the parents, then such situation is called codominance. So answer of this question will be codominance, that is option number 2. Now let's move to the next question, that is question number 4. Question number 4 states that three children in a family have blood types O, AB, B respectively. What are the genotypes of their parents? We have to find out the genotype of parents. This question can be explained by making a Punnett square. We know that genotype IAIB represents blood group AB, IBIEI represents blood group B, IAI represents blood group A, and II represents blood group O. Here it is given that one kid has blood group O. It means one parent. Let's see, this is parent 1, this is parent 2. So both of the parents must have alleles I, because alleles II makes blood group O. So one of the parents must have alleles II. Now next children has blood group AB. It means one parent must have IA and the other parent must have IB. Only then the children will have IA and IB. That is AB blood group. And the next is the one of the children has blood group. It means that one parent must have IB. So let's assume that parent A have IA and parent B have IB alleles. So the parents' genotypes will be IAI and IBIE. I. Now let's see the options. The first option is IAI, IBI. So this is the correct answer. We have explained it here. So answer for this question will be option 1, that is IAI, IBI. Now let's move to the next question, that is question number 105. Question number 105 states that offspring produced from a marriage have only O and A blood groups. The possible genotypes of parents would be here also we have to find out the genotypes of parents. Here given the offspring have 0, O and A blood group types. And we know that O blood group is when one of the both the parents have II alleles, and blood group A will be then when one of the parents have IA, and other description are not given. On the basis of this two blood groups we have to find out the answer. It means that the one of the parent must have II because only A and O blood groups are in the children. So the genotypes of parents will be IAI and II. Now let's see the options. The first option is IAIA and IAI. This is wrong. The second option is IAIA and II is correct, but IAIA is wrong. We have to find it IAI and II. Third option is II and II. This is also wrong. And the fourth option is we have IAI and II. So this answer will be correct. Means the answer for 105 will be option number 4. Now let's move to next question, that is question number 106. Question number 106 states that what is the phenotypic ratio of F2 generation in dihybrid cross? So basically it is a memory based question. You have to memorize the phenotypic ratio of F2 generation in dihybrid cross. And in the phenotypic ratio in dihybrid cross of F2 generation is 9 is to 3 is to 3 is to 1. So here the first option is 9 is to 3 is to 3 is to 1. So option number 1 will be our correct answer. Now let's move to next question, that is question number 107. It says that which one of the following laws of Mendel is based on the inheritance of two genes? We have to find out on the which law was based on the inheritance of two genes. So, the law of independent assortment was based on the inheritance of two genes. Here options are law of dominance, no; law of segregation, no; law of independent assortment. Linkage, no. So the correct answer for this question will be option 3, that is law of independent assortment. Now let's move to next question, that is question number 8, 108. Question number 108 state that Morgan hybridized fruit fly, that is dihybrid cross having characters. Means Morgan hybridized a fruit fly. So what characters the fruit fly has? So Morgan when hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that two genes did not segregate independently of each other, and in the F2 ratio was deviated very significantly from 9 is to 3 is to 3 is to 1. So the characteristic of the fruit fly was yellow-bodied, white-eyed female. That is yellow-bodied, white-eyed female, yellow-bodied, white-eyed female, and the male was brown-bodied, red-eyed. Brown-bodied, red-eyed. It means our answer will be option number 3. Option number 3 is the correct answer. Now let's move to next question. That is question number 109. Question number 109 states that fruit fly, that is Drosophila melanogaster, was found to be very suitable for experimental verification of the chromosomal theory of inheritance by Morgan and his colleagues because we have to find on the basis of what the fruit fly was the best suitable experimental verification for chromosome theory of which was conducted by Morgan. So Morgan worked with the tiny fruit flies, that is Drosophila melanogaster, which were found very suitable for such studies. because they could be grown on a simple synthetic medium in the laboratory. They complete their life cycle in just about 2 weeks. And also, through a single mating, they could produce a large number of progeny. As well as there was a clear differentiation of the sexes, that is, the male and female sexes were clearly distinguishable, and also many types of hereditary variation that can be seen with the low power microscopes. Means to see the types of hereditary variation, high magnified microscope was not required. So these were the reasons they selected Drosophila for their experiment. Now let's see the option. The option is it produces parthenogenetically. So this is not the reason. Here it is not mentioned anywhere. Next, smaller female is easily distinguishable from large female. This is wrong because females are large and males are small. Males are small. Next is a single mating produces two young flies. This is also wrong because a single mating produces many young flies. Next option is it completes its life cycle in about 2 weeks. This is correct. Here we have explained that they complete their life cycle in about 2 weeks. So the correct answer for this question will be option number 4. That is, it completes its life cycle in about 2 weeks. Now let's move to the next question, that is question number 110. This question states that the initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiment carried out in, means on which organism the experiment was conducted initially. So the initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some experiments which was carried out in insects. In fact, the cytological observation made in several insects lead to the development of what concept of the genetic or chromosomal basis of sex determination. So our answer of this question will be insects, that is option number 2. Now let's move to the next question, that is question number 111, that is 111. This question states that 50% of the sperm received a special nuclear structure. Henkin gave a name to this structure as. It was observed by Henkin in 1891 that a specific nuclear structure all through spermatogenesis in few insects can be traced, and it was also observed by him that 50% of the sperm received this structure. But whereas the 50% sperm did not receive it, and Henkin gave this structure a name as X body. So let's see the option. The options are nuclein, no; autosome, no; X body, yes. The Henkin gave name to this structure as X body. And Y chromosome, this is also wrong. So the right option for this question will be option number 3. 50% of the sperm received a specific nuclear structure. Henkin gave a name to this structure as X body. Now let's move to question number 112. Question number 112 is in grasshopper. We have to find out the chromosomal structure of grasshopper. We know that grasshopper is an example of an XO type of sex determination in which the males have only one X chromosome besides the autosomes, and females have a pair of X chromosomes, means females have two X chromosomes. So let's see the answer options. The first option is the male has only one X chromosome. This is the right option. The second option is the female has a pair of X chromosome. This is the right answer. And the third one is the female has only one chromosome. This is wrong because female have two X chromosomes. And the fourth option is both 1 and 2. So the answer of this question will be option 4. That is for both 1 and 2. Now let's move to next question, that is question number 113. Question number 113 states that mark the odd one with respect to true breeding lines. We have to mark the odd one. Here you have to be careful. We have to mark the odd one. So what is true breeding? True breeding means pure line. When a breeding line has undergone several or continuous self-pollination, then it is known as true breeding or pure lines, and it shows the stable trait inheritance and expression for several generations. Now let's read the option. The option is shows the stable trait inheritance. This is right. True breeding line or pure line shows the stable trait inheritance. The next is shows expression for few generation only. This is wrong because it shows expression for several generation, not for few generation. And the third is undergone continuous self-pollination. This is also right. It has undergone several or continuous self-pollination, and we have to find out the odd one. So here the odd one is option number 2, shows expression for few generation. So the answer for this question will be option number 2. Now let's move to the next question, that is question number 114. Question number 114 state that what will be the possible blood groups in children from the parents with B and AB blood groups? Means here the genotypes of parents have been mentioned and we have to find out the possible blood groups in children. So when let's the if the one parent have B phenotype, then its genotypes will be IBA and I. And if the parents have AB blood group, then the genotype will be IAIB. And when we will make a cross between them, then we will get genotypes such as IAI, IBI, IAIB, and IBB. All these IAI represents A blood group, IBI represents B blood group, and IAIB and IAIB represents AB blood group, and IBIB represents B blood group. So the so the kids will have A, AB, and B blood groups. Let's see the option. The first is AO. This is not. The second is AB, AB and O. This is also not the right answer. A, B, AB. This is the correct answer. So answer for this question will be option number 3. Let's move to next question. That is question number 115. In garden peas, starch is synthesized effectively in. We have to find out that in garden peas, starch is synthesized effectively in either heterozygous round-seeded plants, homozygous round-seeded plants, wrinkled seeded plants, or pure and hybrid round-seeded plants. So we know that starch synthesis in pea seeds is controlled by one gene. It has two alleles, that is capital B and small b. Starch is synthesized effectively by BB, that is dominant homozygotes, and therefore large starch grains are produced. And in contrast, BB, that is recessive homozygotes, have lesser efficiency in starch synthesis and it produces a smaller starch grain. It means that capital in BB will produce intermediate starch grain, capital BB will produce large starch grain, and small bb will produce small starch grain. And here the question is, in garden peas, starch is synthesized effectively in. It means it will be synthesized effectively in homozygous round seeded plants. So option 2 is the right option. That is option 2. Now let's move to the next question, that is question number 116. It states that when Mendel self-hybridized the F1 plants, that is RRYY, he found that dominant and recessive traits of one character are segregated in. So, when Mendel self-hybridized the F1 plants, the dominant and recessive traits of one character are segregated in 3 is to 1 ratio. That is, the phenotypic ratio is 3 is to 1 for F1 plant. So the correct answer will be option 4, 3 is to 1 ratio. Now let's move to the next question, that is question number 117. Question number 117 states that heterozygous round and yellow seeded plants were helped and the total of 800 seeds were collected. What is the total number of seeds with first dominant and second recessive traits? So here the first dominant will be round and the second recessive trait will be green seeds. Since out of 16 seeds, 3 are round, 3 are only round and green. Therefore, out of 800 seeds, the ratio of round green plants will be calculated by this formula, that is 3 by 16 multiplied by 800, and it gives 150. So answer for this question will be 150. Let's see the options. The first option is 950, second option is 300, third option is 200, and the fourth option is 150. So correct answer of this question will be fourth, that is 150. Now let's move to the next question, that is 118. Question number 118 states that which of the following statements for the chromosomal theory of inheritance is incorrect? We have to find out the incorrect statement about the chromosomal theory of inheritance. So as per the chromosomal theory of inheritance, pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. And it also, the behavior of chromosome is also parallel to the behavior of genes. The two alleles of a gene pairs are located on homologous sites on homologous chromosomes. Chromosomes as well as genes occurs in pairs. That is, the statement 2 is correct regarding chromosomal theory of inheritance. The statement 3, that is the two alleles of a gene pair are located on homologous sites on homologous chromosome, this is also correct. And the fourth, that is chromosome as well as genes occur in pairs, this is also right. But if we see the first option, it says that pairing and separation of a pair of chromosomes would lead to the segregation of a factor they carried. So this is the incorrect statement because they lead to the segregation of a pair of factor and not the just a factor. A pair of factors they carried, it is segregated. So answer for this question will be option number 1. That is option number 1 is incorrect regarding the chromosomal theory of inheritance. Now let's move to the next question. The next question, that is question number 119. It says that experimental verification of the chromosomal theory of inheritance was given by. Means we have to find out the name of the scientist who has done the experimental verification of the chromosomal theory. It is a memory based question and you have to memorize this fact. And the fact is that experimental verification of the chromosomal theory of inheritance was given by T.H. Morgan. So the third option, that is T.H. Morgan, is the correct answer for this question. Now let's move to question number 120. Question number 120 states that fruit flies are one of the best materials of genetic studies because all except means we have to find out the one option which is not best material for the genetic studies. So we know that the fruit flies, we have also explained in the other question that fruit flies are ability to grow on simple synthetic medium in the laboratory. Fruit flies can easily be grown in a simple synthetic medium. The second option is short lifespan. This is also correct because they have a lifespan of 2 weeks. Now, the next option, that is option third, is production of a large number of progeny in each mating. This is also correct. They produce many progeny in single mating. And the next option is presence of few externally visible and identifiable contrasting traits. So this is not correct because there are many externally visible and identifiable contrasting traits are present in fruit flies. So all are true or correct except option number 4, and we have to choose the wrong statement about this. So the answer will be option number 4. Now let's move to question number 121. It states that generation of non-parental gene combination is termed as. So you have to memorize this also. The non-parental gene combination is known as recombination, and it is produced by crossing over. That is the exchange of genetic materials between non-sister chromatids. So generation of non-parental gene combination is termed as recombination. That is option number 3. Here it is given recombination. Now let's move to the next question, that is question number 122. It states that individuals having homomorphic sex chromosome produce. So what is homomorphic sex chromosome? So homomorphic sex chromosomes means similar types of sex chromosome is present in any individual, like in human females we have XX sex chromosome. So such type of chromosome is known as homomorphic sex chromosome. An individual that having homomorphic sex chromosome presents produce only one type of gametes. It produces only one type of gametes. Let's see the option. The options are only one gamete in the complete lifespan. It is wrong. Not it not produces only one gamete. Next is one type of gametes. Third option is no gametes and two types of gametes. The answer is here clearly written one type of gametes are produced. So answer will be option number 2. One type of gametes is present. Now let's move to the next question, that is question number 123. This question states that which of the following phenomena leads to variation in DNA? So options are, the first option is linkage mutation, second option is recombination linkage, third option is mutation and recombination, and fourth option is aneuploidy and linkage. So let's understand the meaning of these terms. So linkage means the physical association of genes on a chromosome. It means they are linked. Second is mutation. Mutation means sudden or abrupt change in genetic material. Means it can cause variation in DNA. The next option is recombination. Recombination means non-parental combination. Non-parental combination will lead to variation in DNA. Variation in DNA. And the next term is aneuploidy. Aneuploidy means presence of an extra or abnormal number of chromosomes. Means presence or absence of chromosomes, which leads to the abnormal number of chromosomes. It also changes the chromosome, but it can't change the nature of DNA. So here we have to find out the phenomena which leads to the variation in DNA. So we have found here only two phenomena, that is mutation and recombination leads to the variation in DNA. So let's see the options. Option 1 is linkage mutation. This is not linkage, do not create variation, mutation does. Second option is recombination, linkage recombination does, but linkage does not, leads to variation in DNA. And the third option is mutation recombination. Here, both the phenomena creates variation in DNA. And the fourth is aneuploidy linkage. Here, linkage and aneuploidy both do not create variations in DNA. So, answer for this question will be option 3, mutation and recombination. Now, let's move to the next question, that is question number 124. It states that single cell anemia disorder arises due to. We have to find out the reason why single cell anemia disorder rises. So you know that single cell anemia is a genetic disease, and it is basically an autosome linked recessive trait disease that can be transmitted from parents to the offspring when both parents are carrier for the gene. If both parents are not carrier of the gene, then this gene will not be transmitted. And the defect is caused by the substitution of glutamine by valine. Means here you can see the image, one of the bases that changes here. CTC changes into CAC and GAG changes this to GOG. That is glutamine is substituted to valine. That is, it means one, that is single base substitution of DNA is taking place here. So, the reason of why sickle cell anemia disorder arises should be the change in the single base substitution of DNA. Now, let's read the options. The options are duplication of a segment of DNA. No, this is not right. Substitution in a single base of DNA. Yes, this is right. Here we have explained that glutamine is substituted by valence, means there is a single base substitution takes place in DNA. Third option is deletion of a segment of DNA. This is not right. Duplication in a base pair of RNA. This is also not right. So the correct option for this question will be option number 2. Now let's move to the next question, that is question number 125. Question number 125 state that cystic fibrosis, myotonic dystrophy and thalassemia are. We have to find out what kind of these diseases are. Options are chromosomal disorders, autosomal recessive disorders, Mendelian disorders, or autosomal dominant disorders. So the correct answer for this question will be Mendelian orders. That is option 3. Why? Because Mendelian orders follows the Mendelian principle of inheritance. Only Mendelian disorders follows the Mendelian principle of inheritance, and hence they are known as Mendelian disorders. Some other examples of such diseases are color blindness, sickle cell anemia, hemophilia, etc. So answer for this question will be option number 3. Now let's move to the next question, that is question number 126. Question number 126 states that which of the following traits shows transmission from carrier female to male progeny? And the options are autosomal dominant, X-linked recessive, Y-linked recessive, and X-linked dominant. So answer of this question will be X-linked recessive. That is option number 2. Because in X-linked recessive, the transmitted from unaffected mother to the male offspring. That is X-linked recessive gene. Here you can see the mother is affected, means mother is the carrier. She is not affected and the father is normal. And the male, one of the male is normal where other one is affected. And the question is which of the following traits shows transmission from carrier female to male progeny? Here you can see this is a carrier female and the disease has been transmitted to the male progeny. So the answer will be X-linked recessive. Now let's move to the next question, that is question number 127. This question states that phenylketonuria is an inborn error of metabolism that is inherited as. We have to find out in what way this phenylketonuria disease inherited. That is, it is autosomal recessive trait, it is sex-linked dominant trait, or it is X-linked recessive trait, or it is autosomal dominant trait. So basically, phenylketonuria or PKU is an autosomal recessive disorder. Here, the affected individuals lacks an enzyme that converts the phenylalanine into tyrosine, and as a result, this phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Hence, the disease name is phenylketonuria. So, the answer of this question will be, it will be inherited by autosomal recessive disorder. Autosomal recessive trait means this option is the correct answer of this question, option number 1. Now let's move to the next question, that is question number 128. Question number 128 states that the affected individuals are short stature in disorders. Means we have to find out in which disorder the affected individuals are short statured. Here options are, the first option is Turner syndrome, phenylketonuria. Second option is Down syndrome, Turner syndrome. Third option is Klinefelter syndrome, Down syndrome. And fourth option is Turner syndrome, Klinefelter syndrome. So we know that Individuals are affected and short-statured in disorders like Down syndrome and in Turner syndrome. The individual affected by Klinefelter syndrome are masculine, perfectly developed. So the answer will be the option which has Down syndrome and Turner syndrome. Let's see the option. The first option is Turner syndrome, phenylketonuria. It is not right. Second option is Down syndrome and Turner syndrome. So this the option 2 is the correct answer. Now let's move to the next question, that is question number 129. Question number 129 states that which of the following disorder is seen in human females only? We have to find out which disease is seen in human females only. So answer for this question will be 1. That is option number 1, Turner syndrome. Turner syndrome is basically monosomic of X chromosome, means one chromosome, X chromosome is absent. And such disease is known as Turner syndrome, and can be seen in human females only because it is related to X chromosome. Now let's move to the next. So answer of this question will be option number 1. Now let's move to the next question, that is question number 130. It states that thalassemia and sickle cell anemia are caused due to a problem in globin molecule synthesis. Select the correct statement. We have to select the correct statement. Here it is talking about thalassemia and sickle cell anemia. So thalassemia is basically a quantitative problem of synthesizing too few globulin molecules, whereas sickle cell anemia is a qualitative problem and it's synthesizing an incorrectly functioning globule. It synthesizes incorrectly functioning globulin. So difference between them will be the first option is both are due to qualitative defect. This is wrong because one is due to qualitative effect whereas another one is due to quantitative effect. The second option is both are due to quantitative defect in globin chain synthesis. This is also wrong. Third option is thalassemia is due to less synthesis of globin molecule. This is correct. Thalassemia differs from sickle cell anemia in that thalassemia is a quantitative problem and it is synthesizing too few globin molecules. It means it is synthesizing less globule molecules. And the fourth option is sickle cell anemia is due to quantitative problem. It is also wrong because sickle cell anemia is a qualitative problem. So answer for this question will be option 3. Thalassemia is due to less synthesis of globule molecules. Now let's move to next question, that is question number 131. Question number 131 says that the movement of a gene from one linkage group to another is called. It is a different definition type of question. You have to learn this. Translocation is the illegitimate crossing over between non-homologous chromosomes. Hence, the movement of a gene from one linkage group to another is called translocation because it is an illegitimate crossing over between non-homologous chromosomes. So, answer for this question will be option number 4. Now, let's move to the next question, that is question number 132. This question states that which one of the following is the most suitable medium for the culture of Drosophila melanogaster? The options are moist bread, agar-agar, ripe banana, and cow dung. So we know that for the culture of Drosophila melanogaster, one of the most suitable medium is ripe banana. Why? The reason is that ripe banana is rich in carbohydrates, means it provides sugar for the Drosophila and also yeast is present because wherever there is fermentation, there is yeast. And sugar and yeast constitutes an important part of their diet. Means sugar and yeast is an important part of Drosophila's diet. So where these two are present, that will be the best or you can say the most suitable medium for their growth. So here the option 3, that is ripe banana, will be our answer. Option 3. Now let's move to the next question, that is question number 133. Question number 133 states that phenotype of an organism is the result of. So we know that the phenotype is the observable traits, that the traits which can be observed, or physical appearance, physical characteristics of an organism. And it is affected both by genetic constitution as well as the environment around which the individual is living. Hence, a phenotype is the result of genotype as well as the environment interaction. Let's see the option. The option first is mutation and linkage. This is wrong. Option 2 is cytoplasmic effects and nutrition. This is also wrong. Option 3 is environmental changes and sexual dimorphism. It is also wrong. And the third option is genotype and environment interaction. So this is the correct option. That is option number 4 is the correct answer of question number 133. Now let's move to the next question, that is question number 134. Here it is given that how many different kinds of gametes will be produced by a plant having genotype A, B, C. So this we have to find out types of gametes produced by such type of genotypes. So to calculate the total number of gametes that are produced by this particular genotype can be calculated by a formula, and the formula is 2 to the power n, where n is the number of heterogeneous alleles that are found in genotype. Here we have capital AA, capital B, small b, and CC genotype. Means here we have only one heterozygous allele. So the value of n here is 1. And if we put this value in the formula 2 to the power n, then it will give 2. It means that two types of gametes, that is ABC, gametes A, B, C, and gametes A, B, A small b, C, will be produced by plant having genotypes capital A, capital B, small b, and capital CC. So answer for this question will be option number 4, that is 2. Now let's move to the next question, that is question number 135. Question number 135 states that a man and a woman who do not show any apparent signs of certain inherited diseases have 7 children, that is 2 daughters and 5 sons. Three of the sons suffer from the given disease, whereas none of the daughters are affected. Which of the following modes of inheritance do you suggest for this disease? And the options are autosomal dominant, sex-linked dominant, sex-limited recessive, or sex-linked recessive. So answer will be our option number 4, sex-linked recessive. Why? Because sex-linked recessive diseases only affect sons and not daughters. And the reason is son. Here you have also here you can also see that out of 5 sons, 3 sons are affected and 2 sons are not affected by the disease. The sons who are affected, it means that the son who are affected must have inherited the affected X chromosome, that is XC, from their carrier mother. And if the mother is carrier, then this disease will not be transmitted to daughters. Here you can find that the father is healthy and father's genotype is XY, whereas mother is the carrier of the disease and mother's genotype is X and the mother's genotype is XCX. So this is in the case of sex-linked recessive diseases. Hence the option will be option number 4. Now let's move to the section B, and the question number 136. The question number 136 states that a woman with normal vision but whose father was colorblind marries a colorblind man. Suppose that the fourth child of this couple was a boy. This boy means we have to find out the boy is whether normal vision or a colorblind. So if a woman whose father was colorblind, it means she must be a carrier. So genotype of the mother will be XXC, and he marries a color blind man. It means the genotype of the man will be XC by, and when we will cross, it will produce a carrier daughter, and a color blind daughter. And it also produces a normal son and a color blind son. So the chances of the fourth son will be either normal, means the son will maybe normal vision, or he may be color blind. Let's now check the options. The first option is must have normal color vision. This is wrong because the sun may be color blind also. Second option will be partially color blind since he is heterozygous for the color blind mutant allele. This is totally wrong because one individual cannot be a partially color blind. He will be either color blind or normal vision. The third is must be color blind. This is also wrong because sun may be there is possibility of normal vision also. The next option is maybe color blind or maybe of normal vision. So this is the correct option. This option number 4 is the correct answer for this question. Now let's move to the next question, that is question number 137. It says that hemophilia is more commonly seen in human males than in human females because we have to find out the reason why hemophilia is common in human males rather than human female. So we know that this disease hemophilia is due to an X-linked recessive mutation, and females will be affected only when she inherits two X chromosome, whereas male is affected by inheriting only one X chromosome. So if a mother is a carrier for this disease, then there is high chances that the son inherited the affected chromosome of the mother, and then he will be affected by this disease. So this is the reason why the human males are commonly seen hemophilic, whereas human females are not. And the options are, the first option is this disease is due to an X-linked dominant mutation. This is wrong because this disease is due to X-linked recessive mutation. The second option is a greater proportion of girls die in infancy. This is also wrong. No such data is available. Third option is this disease is due to an X-linked recessive mutation. So this is the correct option. And the fourth option is this disease is due to bi-linked recessive mutation. This is also wrong. This is X-linked recessive mutation. So the correct answer for this question will be option number 3. Now let's move to the next question, that is question number 138. Question number 138 states that in order to find out the different types of gametes produced by a pea plant having the genotype AABB, it should be crossed to a plant with the genotype. So, whenever we need to find out the different types of gametes, we can cross the plant with a homozygous recessive, that is AABB. We will cross with this type of genes, and such a cross is known as test cross. In fact, the test cross is given name test cross because it determines the types of gametes. So the answer for this question will be AABB, small AABB, recessive for both genes. Option number 4 is the correct answer. Now let's move to the next question, that is question number 139. Question number 139 states that chromosomal disorders arise due to, means we have to find out the reason of chromosomal disorders. So let's read the options. The first option is absence of one or more chromosomes. This is right. Chromosomal disorder is due to the absence of one chromosome or due to the presence of chromosomes, or it may be due to the abnormal arrangement of one or more chromosomes. Next option is excess of one or more chromosome. This is also correct. Third option is abnormal arrangement of one or more chromosome. This is also correct. These all the three reasons of chromosomal disorders. So, and the fourth option is all the above. So, our answer will be option fourth. That is all the above. Fourth option, all the above. Now, let's move to the next question, that is 140. Which one is the incorrect statement with regard to the importance of pedigree analysis? So we know that pedigree analysis is used to study the inheritance of characters within the family, and it does not explain the nature of the DNA. It is usually, or it is only help to study the inheritance of characters. So let's read the options. The options are: it helps to trace the inheritance of a specific trait. It's right. It confirms that DNA is the carrier of genetic information. This is not right because it does not explain the nature of the DNA. It only explains the inheritance of characters. The third option is it helps to understand whether the trait is dominant or recessive. Yes, it helps in understanding the dominance or recessiveness of a character. And fourth, it confirms that the trait is linked to one of the autosome. It also explains this, but it does not confirm that the DNA is the carrier of genetic information. And we have to find out the incorrect statement regarding pedigree analysis. So correct answer for this question will be option number 2. Now let's move to the next question, that is question number 141. It states that Mendel's principle of segregation means the germ cells always receive. So Mendel's principle of segregation states that the alleles of a pair segregate from each other in such a way that a gamete receives only one of the two genes, that is, one of the paired alleles. During segregation, the gamete receives only one of the two genes. Now let's see the options. The options are one pair of alleles, this is wrong. One quarter of the genes, this is also wrong. Third option is one of the paired alleles, this is right. Here we have mentioned that it receives one of the paired genes, paired alleles, or only one of the two genes. And the fourth option is any pair of alleles. This is also wrong. So the right answer for this question will be option number 3. Now let's move to the next question, that is question number 142. It states that the color blindness is more likely to occur in males than in females because we have to find out the reason why color blindness is more commonly found in males rather than females. So we know that color blindness is also an X-linked recessive disorder like hemophilia, and genes for characters are located on X chromosome, and the females will be affected only when it inherited 2X chromosome, whereas male is affected by inheriting only 1X chromosome, and that's why it is common in males. Now let's check the options. The first option is the Y chromosome of males has the genes for distinguish colors. This is wrong because X chromosome have these properties. Second is genes for characters are located on the X chromosome. This is the right answer. The genes for the characters are located on the X chromosome. Now let's read the other option as well. The other option is the trait is dominant in males and recessive in females. No, this is also wrong. And the fourth is none of these. So the answer for this question will be option number 2. That the genes for characters are located on the X chromosome. Now let's move to the next question, that is question number 143. It says that a polygenic trait is controlled by three genes, that is A, B, and C. In a cross AABBCC cross AABBCC, the phenotypic ratio of the spring was observed as 1 is to 6 is to x is to 20 is to x is to 6 is to 1. What is the possible value of x? We have to find out the value of x. So we know that in polygenic inheritance, especially in the case of three genes, the phenotypic ratio of the offspring is 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1. You have to memorize this phenotypic ratio in case of polygenic inheritance, and by comparing this ratio with this that is given in question, we can find out the value of x, and by comparing this we find that the value of x should be 15. Here 1 is to 6, 1 is to 6 is to 15 is to 20 is to 20 is to 15 is to 6 is to 6. So answer for this question will be option number 3. that is 15. Now let's move to the next question, that is question number 144. Question number 144 states that if two pea plants having red, that is dominant colored flowers with unknown genotypes are crossed, 75% of the flowers are red and 25% are white. The genotypic constitution of the parents having red colored flowers will be. We have to find out the genotypic constitution of parents. If 75% of the flowers are red and 25% of the flower are white. So we can explain it by the performing this cross. Let's assume that both the parents are heterozygous. So when we cross between two heterozygous parents, it gives this ratio, that is two red and one white. It means that 75% are red and 25% are white. That the phenotypic ratio is 3 is to 1. So the answer for this question will be both heterozygous. Here we have crossed both heterozygous parents. So answer will be option number 3. Now let's move to the next question, that is question number 145. Question number 145 states that in plants, yellow seeds are dominant to green. If a heterozygous yellow seeded plant is crossed with a green seeded plant, what ratio of yellow and green seeded plants would you expect in F1 generation? We have to find out the ratio of yellow and green seeded plants in F1 generation. So when we cross a heterozygous yellow seeded. This is a heterozygous yellow seeded plant with green seeded homozygous plant. Then we will get this type of progeny, that is YY is to YY. Means the ratio is 1 is to 1. Means 50% will be yellow seeded and 50% will be green seeded. So answer for this question will be 50 is to 50. Let's check the option. The options are 3 is to 1. No. 50 is to 50. Yes, this leads to the phenotypic ratio 1 is to 1. 9 is to 1. This is wrong. 1 is to 3. This is also wrong. So correct option for this question will be option number 3. Now let's move to the next question, that is 146. This question states that if a color-blind man marries a woman who is homozygous for normal color vision, the possibility of their son being color-blind is. So we know that color blindness is an X-linked recessive disease and it shows crisscross inheritance. It means that a color-blind man and a normal man cannot have a color-blind son because the son receives X chromosome from his mother and mother is a normal vision means he has XX chromosome. Hence, there is no chances that the son will be colorblind. So the answer of this question will be 0. Means let's check the options. The option is 0, 0 0.5, 0 0.74, 1. So option 1 will be correct answer. There is no chance that sun will be colorblind. Now let's move to the next question, that is question number 147. It states that in a dihybrid cross there are 16 squares. How many different types of genotypes and phenotypes are formed respectively? So you have to memorize this, that in a dihybrid cross where there are 16 squares, there are 9 types of genotypes. RRYY, RRYY, these are the 9 types of genotypes and 4 types of phenotypes. That is round yellow, round green, wrinkled yellow and wrinkled green. So answer of this question will be 9 and 4. Let's check the option. The option is, first option is 9 and 4. So this is the correct answer of our question, option number 1. Now let's move to the next question, that is question number 148. It states that in a cross between capital ABB and AABB, the ratio of genotype in F2 generation between AABB, this would be, we have to find out the genotypic we have to find out the ratio of genotypes in F2 generation. This could be explained by this Punnett square. When we made a cross between this, we find a ratio of 2 is to 4 is to 2 is to 1. That is AABB is 2. Here you can find AA, 1 AABB and 2. Like this, you can find AABB 4. Let's find out AABB here 1, AABB here 2, and AABB here 3, and AABB here 4. Next is AABB. Here you can find AABB 1 and AABB 2. And the next is small AABB. There, small AABB. It is 1 in number. So ratio will be 2 is to 4 is to 2 is to 1. So let's check the option. The first option is 9 is to 3 is to 3 is to 1. This is wrong. Next option is 2 is to 4 is to 2 is to 1. This is right. So answer for this question will be option number 2. Now let's move to the next question, that is question number 149. Question number 149 states that in the given pedigree, what is the mode of inheritance? In this given pedigree, we have to find out the mode of inheritance. So, the mode of inheritance is autosomal recessive. Here, the mode of inheritance will be autosomal recessive. And the options are autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. So, answer will be option number 2, autosomal recessive. And why? Because autosomal recessive pedigree tends to show fewer affected individuals. It shows only few individuals are affected by this and are often described as skipping generation, means in one of the generation is unaffected by this. Here you can see that this is the first generation, this is the first generation, this is the second generation, and this is the third generation. Individuals are affected in second and third generation, but the first generation, that is parents, are not affected. And in autosomal recessive type of pedigree, they shows skipping generation. Means this one generation has been skipped. Hence, this pedigree shows the mode of inheritance which is autosomal recessive. Now let's move to the last question. And the last question, that is question number 150, is in garden pea, the yellow color of cotyledons is dominant. The yellow color of cotyledon is dominant over green, and the round shape of seed is dominant over red. Means yellow color is dominant over green and the round shape is dominant over wrinkle. When a plant with yellow and round seeds is crossed with a plant having yellow and wrinkled seeds, the progeny shows segregation for all four characters. Means all four characters are segregated. The probability of obtaining green round seeds in the progeny of this cross, we have to find out the probability of green round seeds. So here it is written that yellow color cotyledon is dominant. So we will represent it with capital Y, whereas round shape is dominant, so it will be represented by capital R, capital R. Here the green color is recessive, so it will be represented by small y, and wrinkle shaped is recessive, it will be represented by smaller. And the question says that when a plant with yellow and round seed, means yellow and round seed, this will be the genotype of the plant. One plant is crossed with a plant having yellow and wrinkle seed. Yellow and wrinkle seed. So this could be the genotype of the other plant. And if the progeny shows segregation in all four characters, what will the probability of green round seeds? So we will make a Punnett square like this. These are the gametes which will be formed because of segregation of all the four characters. And when we will cross them, we will find here total number of progeny. Total number of progeny here is 16. Total number of progeny is 16. And the genotypes for green rounds, here we will find 1, 2 and 3. Only 3. Here you can see this one is the green round. This one is the one green round. Another is RRYY. Here is the green round. There is the second green round, and the next is RRY. This is RR small YY. This is also green round. RRYY. RR, sorry. This is green round. This is green round. So here we can find only three green round. It means that that 3 out of 16 progenies are green round. So if we find out the probability of obtaining green round seeds, we will find 3 by 16. Means 3 out of 16 are green round. So answer for this question will be 3 by 16. Let's check the option. The options are 1 by 8, this is wrong. Second option is 1 by 16, this is wrong. Third option is 1 by 4, this is also wrong. But the fourth option, that is 3 by 16, is the right answer. So answer for this question will be option number 4. Hope you have liked the question as well as with solution. Thank you.